ourselves to record.
All right.
All right, so it says it's recording.
I see. Yeah.
So I'm here with Bernie Sanders and
Most
my
importantly.
my long term pal Sammy Verzino from back in Holy Cross High School, which we were just,
Yes.
and I'm Dan, which we were just talking about last night and today with a long conversation about what was it, Catholic schools and private schools and whether
Yeah.
they're
The
better than public schools.
Yeah, or they're just. I guess it started off with just mostly. I feel like a questioning of their viability in
Mm,
a post-pandemic and even pre-pandemic era. I feel that was, and you can say that about a lot of places like retail, schooling in general. But I think the parochial high school
yeah,
foundation is I think it's been crumbling for a while in certain more disenfranchised areas, and I think that this has really just kind of the past years really just kind of started to put the nails in the coffins. Um,
yeah. Although I've heard
but
some people send their kids to private, like have been sending their kids to Catholic school because they were open, at least in Waterbury,
sure,
when public schools were not.
sure, yeah. Um, well, I mean, I think for our audience here, we were discussing it. It was Daniel here, myself, and our good friend Adam Lawley, and we were getting into the ins and outs of schooling in general. It really didn't. The trajectory didn't start that way. It was mostly about a high school in our town, our shared upbringing,
Mm-hmm.
um, is shutting down after probably a
It's been
hundred
open
years
for
at
not a
this point.
hundred. Sacred Heart was
Close,
like, because my grandma was
post war.
remembers when they made Sacred Heart. But it's been
Okay,
around a while, like maybe
okay,
70 years.
okay. Yeah, I guess I was exaggerating with 100, but we're getting up there
Long,
too. Because,
yeah,
well, you got to think there was. I never saw it, but I heard stories, at least from my grandparents, about Waterbury Catholic. I don't know if that name rings a bell to
yeah.
you.
Catholic High. That's where my
Catholic
grandma
High.
went to high school, and then they merged, I think,
And
with Sacred
yes,
Heart.
and correct me if I'm wrong, that ended up becoming Sacred Heart.
Yeah.
So yeah, I mean, my whole thing was just that I don't think that you can continue to raise tuition steadily throughout the years
Right.
simply to keep up with inflation while also not getting any net benefit. And I think say in some cases having a decline in your what you provide to these
Mm-hmm.
children educationally. I don't think that, and that's not to say that I thought that like I don't think that you need to have a master's degree in teaching in order to formally educate kids, right?
Mm-hmm.
I But what I do think is that in a system where the only regulator is yourself, it's easy to slip through the cracks. And I think that that's what I saw in our alma mater, and I can only imagine that that's what Sacred Heart was going through as well. So
Yeah, I mean, we could definitely talk about that for a
yeah, that's
while.
a
I
different.
didn't even finish reading the comments in that
Yeah,
conversation.
it was it wasn't even like a big thing. It was mostly us just kind of talking about it. I think Lolly thought we were coming at him for a moment because I was disagreeing with
Yeah.
his point jokingly by saying we should revert to monkeys. But
I
uh,
just came in with like the school
you came
sucks
in with
comment
yeah,
and
school
just
sucks.
disappeared.
What did you say? You said yeah, you said
Eat
school
trash,
sucks.
be free.
Yeah, eat trash, be free, and then just
Education is indoctrination.
you basically like you lit a match, threw it into the chat room, and bounced for a couple hours.
I didn't think that anybody was going to be upset
Well,
by it.
yeah, that was I didn't either. That's why I posted a two monkey memes. POV, you are banana.
So anyway, I don't know. I thought that might be a nice segue into our kind of
Yes, academic talk.
academic discussion. But you said you wanted to start by asking me a question.
I
We can
did.
start there.
Well, let me. I guess it's kind of a moot point at this point, but I'll also say I know you introduced me. Hello, I'm Sam Verzino.
Mm-hmm.
And my question to you, Dan, is what is the name of this podcast, if one
Career.
exists?
Yeah, it's gonna be called Career Day. This would probably be
Okay.
episode 1. Did I not tell you that?
I don't. I I don't know. I'm gonna wager on the side of no, just so I don't sound stupid and say yes and
So
forgot
yeah,
about it. But I
it'll be
like
called,
Career Day.
yeah, with the theme of people talking about their careers, but I think it'll be much more in depth and hopefully interesting than like your traditional high school career day, where like maybe one
Sure.
person comes in or
Yeah.
some people talk for 10 minutes. My goal
Yeah.
is to have 1 hour episodes with different people, and like the more
Yeah.
I think about it, the more I realize that like I could have a bunch of different guests. It doesn't have to be like somebody with an extremely weird career. I think
Yeah.
like even a common career will be an interesting guest. So
Yeah. I mean, I think that that's the basis of a lot of those like middle school or elementary school career days when like someone's parent comes in
yeah,
and talks about, their just general desk job or something like that.
but
Um,
today is all about you, Sam. So
oh, I love it.
Sam's career, which is
Hmm.
his first guest on the show, honor. He's a PhD candidate, right, in
Yes.
biology
Yes.
at is it St. John's University
Yes.
in New York
Currently
City.
at St. John's. Yes.
And how long have you been in the PhD program?
So this is my second full year as a doctoral candidate. I
Okay.
started my grad school experience in 2017, fall of 2017 at NYU, going for my master's degree.
Mm-hmm.
So I had originally intended to continue on through there at the PhD program, um, but when it came down to it, a lot of it did come down to finances for my switch, the reasoning for my switch, as I started my PhD course. But we can get into that, later on or whatever.
Mm-hmm.
Um, but yes, currently at St. John's, second year as a quote unquote doctoral candidate. Um,
Well, isn't that what it is?
Sorry. Yeah, it is. I mean, that is what it is. I say second year because it's like, this is my second year with, my transcript saying doctoral
Okay.
candidate, but
And
it's also
before that, you were a
but before that,
master's, right?
yeah, but before grad school is like, when it comes to science like that, you're still, master's is kind of like baby's first,
Mm-hmm.
uh, you know, foray into lab work, et cetera, and having a
Sure.
PI and teaching, et cetera, and getting a foothold and all that. So overall, fourth year overall, second year as a doctoral candidate.
So when you enrolled at NYU, do you enroll in
Mhm.
the master's program or like the PhD program, or do they not even distinguish at that point?
So they do distinguish. They distinguish as you can apply to either or both. Actually, I
Mm-hmm.
applied to their PhD program. I actually originally didn't get into their PhD program when I applied in my first round of applications. When I was originally applying to PhD programs in from fall 2016 to spring 2017.
Mm-hmm.
Um, but when I got my letter from NYU, they basically were like, we can't offer you a position in our doctoral program right now. However, we would love to have you for our master's program. So it actually came down to I was either going to do I got into the PhD program at Rutgers at the time.
Mm-hmm.
So it came down to going between that or NYU, and the difference was that Rutgers didn't have I didn't want to go there very much anyway, to be honest. Commute
Okay.
and I didn't think they had, from what I had talked to the professors I talked to in my interview rounds, I didn't really connect with anybody in that type of way. Um,
Mm-hmm.
Nothing against the university, great university. They just actually merged with NJIT, New Jersey Institute of Technology, several years ago. So
Mm-hmm.
um, they're putting a lot of their weight behind their science and technologies programs up at Rutgers in Jersey. Great program. Just wasn't for me at the time, and also the money constraint was absolutely an issue because I was not offered um, a stipend in as well as tuition remission. Whereas with NYU, it was the 2 years with a possibility for an application-based stipend position, and I took the gamble. I rolled the dice and went with NYU at the
So
time.
yeah, that was a question I had about finances because I
Yeah.
think a lot of people before undergrad don't realize that there's funding like for PhDs in grad school. A lot of people
Yeah.
just see grad school as something they're going to be in tons of debt for, and they don't want to do that. But
Hmm.
could you talk a bit about like your experience with funding and like how easy it is to get funded for grad school?
Yes. So it's a good question. I can only obviously speak to my experience, and I will make the distinction between it depends on what program you're going into for the most part. So if you're going into a more humanities-based position, like you want to get your PhD in history or literature or even a psychology or something like that, many times those programs are not fully funded. You will have to, whether it's a master's or a PhD, you'll often have to take out loans. For PhD programs, probably for the first year or two, if you're in the humanities, before you can get into a lab, or in the case of the humanities, it wouldn't be a lab, it'd be like a department where you are doing TA-ship, and they can give you some sort of tuition remission and a living stipend, basically for your work for the university. Whereas with the hard sciences, more like biology, chemistry, and physics, and pharmaceuticals as well, most of those programs in American universities are subsidized. So if you get into one, you should most likely be getting tuition remission as well as a stipend, so a salary as well. And if you don't get that upon acceptance of the program, I personally would say don't take it, even if you get into the program. It's not necessarily worth it, and you don't want to go into a bunch of debt for something that's statistically a lot of people don't finish.
Yeah. So you got a stipend at NYU right from the
I
get-go?
no, so that was the gamble I was talking about. It's a master's program, so there's no tuition remission, right? You pay the tuition, so I took out a grad loan for that. But as far as a salary is concerned, to support yourself beyond just the loans you're taking out for your tuition, they had master's students in biology and chemistry, et cetera, can basically, it's an application-based position to be a TA. Or like a student adjunct professor, basically. Like the for those of you maybe listening, when you were an undergrad, if you took any undergrad science course, right, like the seemingly too young to be a professor person that was teaching you, like that's me
That
right
was
now.
Sam.
That's me. So for my master's program, I actually applied first semester. I applied to teach a variety of classes, specifically NYU's is called like principles of biology. And it's like basically like your intro courses that you would take as a freshman in a biology
Bio
major.
1. Huh?
Yeah, bio 1, bio 2, right? So I applied to teach those. I actually was able to secure one of the teaching positions, which was great because it's, they paid pretty well and there was only a few of them available. So I got that right after my first semester. So from my second through my fourth semester at NYU, I was teaching courses for a salary. So that was great. So in that case, for me, the gamble paid off. And when I look back in hindsight in general, I don't regret making the decision to do my master's first, despite getting into a PhD program elsewhere. I everything worked out for me, I think, in a lot of ways, and I wouldn't go back and change it if I could, basically.
Yeah, I mean, NYU is also very prestigious, right?
Yeah, I thought it would be great to have that kind of like, not only the experience of being there and being around the great people that are there, but also, yeah, like there's a point to it where you have to, know that you got to market yourself in a competitive field as such, and to have that on my transcript and resume, I think was a good decision and a good long-term benefit for me.
For sure. Yes,
Yeah.
I mean, my impression with funding has been, and this could be inaccurate, that like for most master's programs, they don't fund you.
Yeah,
And for
that's pretty
most
accurate.
PhDs, they do,
Yeah.
even if it's in humanities. But
Yeah, I shouldn't say most. I definitely taking some liberties there. It's not most of the humanities are not going to fund you. It's just that on the whole, you're not going to find many hard science programs that aren't fully funded. Whereas like there are definitely programs throughout universities that you can start your PhD and not really have any type of secured funding. And you kind of have to, in that case, one of the struggles of being a doctoral candidate is you have to do something, right? You have to start making connections or getting a grant or writing to the point where, oh, now you do have funding.
That's
And
crazy to be writing a grant as a student. You're still doing school.
Yes.
So we're already seeing how money has a lot to do with our career choices. Money in
Yes, it does.
school. I will say there are definitely some. If anybody's out there listening, wondering how they can get money, like if you look around, you'll find some opportunities. My friend Jesus is doing his last semester of a master's at Providence College, and they like for this particular master's in higher education. I don't know why this program in particular does it, but they offer full tuition. Uh, they just waive the tuition as long as you work some sort of like basically an academic assistant job, like doing kind of
Mm.
secretarial organizational work. So you just have to pay for your living arrangements or whatever. So that worked out
That's
pretty
great.
well for him. And then actually during the pandemic, since all on-campus jobs were canceled, they pretty much just gave him the money as a stipend because he had already signed on to that deal.
Oh,
So
that's amazing.
yeah, that's just something I always like to tell kids
Hmm.
about is how to get money for education.
Hmm.
Like my own case, I was lucky to get a full scholarship for high school, and then went to a
Hmm.
school where they meet 100% of demonstrated financial need. So it's like you basically do the FAFSA, and I think the school has their own like financial document that you fill out. But
Mm.
then they look basically at whatever money and assets your parents have, and they determine if you whatever your
Sure.
need is, they meet it without loans. So that's
Yeah.
schools that have big endowments tend to be able to do that.
Yeah.
But
Of
where
course.
there's a will, there's a way, right? I feel like that's the
Yes.
take home message.
I think so. I think it's harder than it needs to be a lot of times, especially in
Sure.
America. But I think that there are opportunities that you do need to hunt for, and
Hmm.
apply to certain grants, application-based grants, as you were saying too,
Yeah,
are big.
and it really is kind of the problem I think is worse because it's creates so much inequality, especially like along racial lines, right? People
Yeah.
from low income families, minority families are often the ones that struggle the most to get
Yes.
these expensive education. So I think that's where like people who come from backgrounds like that really would benefit the most from learning about
Hmm.
scholarships and these other ways to finance education. Because
Yeah.
I've worked in like the Waterbury public school system, which is mostly students of color,
Yeah.
mostly like low to middle income families, and
Yeah.
I think very few of them realize that there's a lot of money and grants. Well, maybe not a lot, but a decent amount at least of like money
Yeah.
for school to be had if you're a good student and
Yeah.
if you go
Something to
for it.
strive for. Yeah.
Yeah, but who knows? I don't know if like scholarships and PhD funding is ever going to be enough to create a truly equal
No,
society. I
I
doubt
mean
it.
it's really not. I mean, there's also there's the issue that comes with funding in general, where it's like you kind of, with me, right? Like it's not like you're not going to coast either. It's I get paid and I don't pay tuition, but I'm still not a high earner by any
Hmm.
stretch of the imagination. You barely make ends meet with what you get paid.
Yeah,
So I've
right.
had to do other things to
Oh,
supplement my income.
what sorts of things, Sam?
Subscribe to my OnlyFans. Uh, no, well, actually, I work. I've been doing this for a while since I graduated. I went to Fordham for undergrad, and I've been working with like sports production and sports media there since I graduated. It's just my little side hustle where
Hmm.
I'll do camera work for like basketball and football and softball and baseball in the spring.
Nice.
And like if you end up doing like 8 to 10 games a month, get a little, it's an extra $1,000 in your pocket, which is what you
Hmm.
need to be able to spend money, for me at least, outside once I've paid my rent and my bills. So
Everybody
um,
should have a side hustle. Life
yes,
lessons that we were never taught. Have your side hustle.
yes, and
Um,
and it doesn't have to be a hobby. Don't make all your hobbies side hustles, because at that point you just don't have any hobbies anymore,
are
and we
you
need things.
criticizing? Are you criticizing my podcast idea,
No,
Sam?
no, not at all, not at all. I think a podcast is the perfect overlap of hobby and side hustle. That is, it's the career of champions.
This
Um,
podcast brought to you by Squarespace. Just kidding.
soon,
But
soon you'll
maybe,
be
unless.
yeah, soon you'll be cutting your guest off to talk about me undies for 35 seconds, and
Or my
how
pillow.
my oh no, I might leave the podcast if you start talking about that. No.
So let's get a little bit more into the story of the soon-to-be Dr. Verzino.
Sure.
Did you know what you wanted to do when you applied for a PhD? Like, did you know what kind of research you wanted to do, or what kind of what you wanted to do
Hmm.
after your
Which
master's?
round of applications? The first time or the second time? Because the
You
first time
take
was
it
when I.
as you will.
Okay. Okay. So the first time, no. When I initially applied to programs, I was applying to master's and PhD programs in late
So you
2016,
applied
early
twice.
2017. I did apply twice, but that's I'll get into that. Basically, the round of applications I was previously talking about is the one that I first did, where I had gotten into Rutgers and also NYU.
Yeah.
Um, at that point, I didn't really know what I wanted to do.
You just knew
To be,
biology.
I just knew biology. I had an undergraduate degree in it. I had spent the past few months working in um, at Memorial Sloan Kettering research
Mhm.
labs, the Rockefeller Research Labs in the Upper East Side on 67th. So that was kind of like my off year in between undergrad and grad school was me working full time there and just trying to do applications and
Mhm.
etc. So that at that point, I didn't have the greatest grasp of what I wanted to do as far as my research is concerned.
Mhm.
And I think that a lot of that had to do with not really having as much of a knowledge base. Oh, we got a guest star Thistle here.
Hey, Thistle, right? So
Yes, we've
did
got
you
my pup
know
Thistle.
if you wanted to do microbiome versus macrobiome?
No, see, I did it. I knew I wanted to do something molecular based,
Okay.
not environmental, because I had
Yeah.
flirted with ecology a little bit my senior year in
Right.
undergrad and didn't really find it as fascinating as I thought I would.
Mhm.
So I knew I wanted to go into molecular. At the time, I was working in a C. elegans lab at
Mhm.
Sloan Kettering. So, there was that part of me that was
The
like,
worms, right? The
yeah,
flatworms.
worms. Yeah,
How
flatworms,
big is a C.
nematodes.
elegans?
C. elegans they're not microscopic. You can see them with the naked eye, but they're pretty small. A
Mm.
full grown C. elegans, I think, is half a centimeter.
Yeah.
Um,
It's a
to
very
to
common
observe,
research animal, right? We
yeah,
know a lot about
yes,
we got its genome sequenced many years
yes,
ago, right?
yes, we have actually. Good job.
Why
It
do
is.
people love C. elegans so much to study?
Um, well, actually, that's a good question. So it's a
And it's
very.
not CSEA, it's like C,
Oh
the
yeah,
letter
it's
C dot
the
elegans, as in
yes,
the
the
genus
the
is,
Latin scientific notation,
yeah,
right?
C
C
dot
period
elegans.
elegance, all in italics as well.
You probably
Um,
got some in your body right now.
Maybe, yeah, probably. So the reason they are such a highly used model organism is because, well, several factors. Physically, they're easy to grow, they're easy to maintain. They have a life cycle of about 10 days. They can
Mm.
self-replicate. There
So
are no
they're
female.
asexual reproductive.
There will sort of. So it's funny. There are no female C. elegans, but there are male C. elegans and hermaphrodites.
Oh.
Males can fertilize just as a male mammal or something would, but a
Hmm.
hermaphrodite can self-fertilize and produce. Um, we aren't really.
Self-fertilize, or like so they can either have sex with themselves or with another male.
Yes, yes, they can.
Is
So
that
males
gay?
are. I don't know. I don't
They're
know if
gay
it's
C.
gay.
elegans.
They probably. I would assume they're all kind of gay if they're only dude. It's a bunch of dudes and like a few hermaphrodites, you know. Um,
What do you think
so
about that, Mitch McConnell? Riddle me this.
riddle me this,
We
United
just
States
start
Congress.
writing letters to conservative politicians. Is C. elegans
C.
gay?
elegans gay, fellas. Is it gay to study C. elegans? They can literally fuck themselves.
So,
Um,
so you're studying C. elegans and you are interested in it?
I was interested in the general premise behind it, and this is the thing that I think a lot of people get confused with model organisms, even like undergrads when they see like, oh, you work in a lab with C. elegans, like, oh, why do so many people work with C. elegans? Why do so many people work with Drosophila? Why do so many people
Hmm.
work with mice? Why do so many people work with yeast? And the reason behind that is all four of those things are very common model organisms, but they're model organisms not because somebody one day decided that they're going to be, but because of having the features like what I just said, like with the C. elegans, fast reproduction time, ability to
Hmm.
self-reproduce, easy to store, easy to
Hmm.
manage. They are fully sequenced. Their genome is fully sequenced. We have a full map actually of their neuronal pathways, and we have an actual The thing with C. elegans now, specifically why they're so popular as a model organism and always have been, is because we know every single neuron, and we have a name and a
Mm-hmm.
number for every single neuron that ever develops in the C. elegans lifespan, as well
Wow.
as a specific timing map to know down to like the minute at what point those neurons are going to become fully developed and active. And then at what point they get to basically a programmed cell death or an apoptosis.
Mm.
Um, so that is why they're so used in that sense for eukaryotic organisms and being a eukaryotic model organism, multicellular eukaryotic organisms. We've learned a lot about growth, development, and neuronal pathways through
Mm-hmm.
them. Away and the way that basically neurons interact with each other, and the way that certain proteins develop when you manipulate and inject, etc. Um,
Mm-hmm.
and they're
Right.
also, and I think probably the biggest physical attribute as to why they're a great model organism is that they are transparent. They're naturally
Mm-hmm.
transparent organisms. So when we, and you've probably heard of like a GFP before, right? Green fluorescent protein. It's a
I've
modern,
heard of it. Yeah.
it's probably the most common molecular workhorse in biology, and basically what it is, it's a synthesized protein that we originally synthesized from undersea algae, and it's fluorescent, right? So it glows in the presence of UV light. So basically what I'm saying is we can tag, remember when I said we have all those neurons in a C. elegans, we can tag any of those neurons with a GFP protein and observe its interactions with every other neuron just by going into a dark room and observing a plate full of C. elegans under a UV light microscope.
Mm-hmm.
You know, special techniques, no cross sectioning, no dyeing, no staining, no soaking, any of that. You put a plate of food uh, down for the C. elegans that contains a protein, a modified protein with a fluorescent marker in it.
Mm-hmm.
The worms eat that, it gets naturally integrated into their genome. A day later, you can be under a microscope and observe every movement, every positioning of this neuron, and every basically part of this C. elegans body, just because it's so easy to reproduce. And then they reproduce eggs pretty quickly. You can pick eggs. You can freeze C. elegans eggs pretty much indefinitely. You can freeze a C. elegans egg in a -80 um, super lab freezer, and you can uh, come back 20 years later and bring those eggs up to temperature, and they will hatch into live C. elegans. Um, very, very, very handy, useful model organism. So that's my long-winded explanation about C. elegans. To basically say that it's a model organism, and it got me not interested necessarily in working with them,
Mm-hmm.
but in working in a molecular lab where I was studying protein and RNA interactions.
Yeah.
Um, so I from that point I knew I wanted to get into molecular. I knew I liked the central dogma. I just didn't know
Mm-hmm.
if I wanted to study neuron development, neuroscience. Did I want to study RNA development? Did I want to study protein function? Where was my niche? I didn't really know. So I was just kind of applying there with the knowledge base I had, and hoping that once I got into a program, I would learn enough or get into a lab where I could kind of
Mm-hmm.
experiment with those ideas. And that also led me to my decision to go with the master's program first instead of the PhD. Um,
Mm-hmm.
because I think that if I had gone into the PhD blindly, as blind as I had been at that time, I think I would have wasted a lot of time um, trying to shop around different labs and not really making the progress that I
Right.
should be making over the span of however many years I'm able to attain funding there.
Yeah,
Um,
I've heard from a lot of people that doing like a kind of gap year in a research lab is very beneficial. I mean, not only does
mhm.
it make your application look stronger, but it gives you some more knowledge and clarity about what you might actually like to study as a grad student,
Absolutely.
right? That's actually something
Absolutely.
I'm trying to do again now. It's been a few years since I did undergrad, but I'm trying to get into a lab to do
Hmm.
like some sort of cognitive psychology, probably, because
Oh, that's great. Yeah.
I did a little bit of like that molecular bio stuff
Uh-huh.
in school, but honestly, not as much as like I really should have to work in like a
Sure.
neurobiology lab. So I
Sure.
feel like my my opportunity lies more with like human subjects research and like brain imaging
Sure.
stuff. Even
Yeah,
though
because
I
you're a
think, you know,
neuroscience guy.
well, yeah, but neuroscience often means like what you're talking about, studying
Yep.
C. elegans neurons.
Yeah.
Unfortunately, I think I kind of missed the boat on that one
Mm.
personally, but it is fascinating
Never too late.
about that stuff. Well, I'll put it this way. I've applied to labs like that and like never
Hmm.
heard from them because I think I just don't have the experience that they're looking for.
Sure.
So like I
Yeah.
would have to probably go to a like pay my way through a master's degree in order
Mm,
to get the experience that I need, if
sure.
that makes
Yeah.
sense.
I know what you're
Whereas
saying.
in like human subjects research, I have a lot more experience, so I can
Yeah.
probably get a job in a lab more easily.
Yeah. Your resume looks a little better in that respect. Um,
Yeah. So what you do in undergrad does matter to an
it
extent.
does. It absolutely does. And I think that I learned that a lot too because I learned it. I learned it in a bad way too, where I got into my master's program and it was hard when I started, and it was difficult to keep up with the coursework, and I think it was the I really had to apply myself in a way that I thought I did in college, but um, you couldn't really coast through what I was doing at that time, and I think that
Hmm.
in my 2 years of classes as a master's student, I learned so much about biology between just 2 years of working in a lab, teaching undergrads, and also learning higher level molecular biology stuff,
Yeah.
as well as techniques in lab courses. Um, I really like that stuff that had kind of just been swimming around in this like primordial soup of like knowledge, kind of
Yeah.
really got solidified in a lot of ways and really helped me to hit the ground running when I actually did get into a doctoral program.
Speaking of primordial soup,
Hmm.
let's get into your current research, which is really, I think, the
Yeah,
most cool thing that I'd like to talk about. So, what are you
sure.
researching? And are you doing
So
a dissertation kind of? This is like your own research project?
yeah, so I'm currently at the point where so I just talks about COVID aside, I was kind of in a precarious position where last year, so fall 2019, spring 2020 was my first year at St. John's, first year as a doctoral candidate, right? So
Mm-hmm.
um your second semester is when you should start to shop for a rotation for a lab. And obviously, of course, come around this time last year during my second semester, we everything got boarded up,
Mm-hmm.
so to speak, around the world. So being in a lab, let alone shopping for one, was a non-option for me.
Mm-hmm.
So I got really delayed until the summer with doing a rotation. So I had to delay my first rotation until late spring last year, early
Okay.
summer. So I did two separate rotations. I did one in a lab that was the head of the department, where we spent a couple weeks working on basically what I kind of did in my research lab at MSK as a lab tech, working with C. elegans and also Drosophila to basically, I was just kind of seeing what his project was like, and they
Mm-hmm.
were working on like neuronal development, et cetera, too. Um, but my second rotation was the one that I want to talk about because it's actually the lab I committed to um,
Mm-hmm.
earlier, or I guess late last year. Um, it's a new professor by the name of Javier Juarez. He actually went to the University of Barcelona, but he was a postdoc at
Barcelona.
Harvard.
Yeah.
Barcelona, sorry, yeah. And he was a postdoc at Harvard for the past couple of years, and he is very much into. Um, he's an RNA protein scientist. However,
Okay.
he is into biotech, right?
Mm-hmm.
So his whole thing is he's actually been working on patents for the last couple of years, or trying to work on a public patent for this biosensor technology that really is like an early detection of oral squamous cancers. And
Okay.
hopefully that can be extrapolated to other pathological diseases,
Mm-hmm.
where basically the concept there is, in layman's terms, is that there's a hallmark protein that is in oral squamous cancer, any oral cancer, that like basically
Mm-hmm.
99.99% of cancers have this production of this protein in saliva. If you have
If it's an oral
if
cancer.
you if you have an oral cancer, and you're 99%, like 99.99% of studied cases of oral cancer have a significant synthesized amount of this specific protein
Does the protein have a
in
name?
yes, it's called PIP high, PIPHY.
We hate
And
it.
there's
Boo.
we hate it. There's
Cancer.
also a couple more. There's one called AMAR, AMA capital R. There's one SATA, like SAT, and
And
then
how do they name these? They're all acronyms,
SIAP.
right?
So yeah, they're acronyms. It's basically like the first, I forget the actual dogmatic way you name protein, but it's like the first three letters of the full of the organism it's from,
Mm.
along with, and the capital letter at the end is, I'm pretty sure the capital letter at the end is like the first letter of the type of protein it is.
Okay.
So for example, AMAR, or sorry, SATA, is from a, SAT is from the organism it's from or whatever, and it's like a sat, I don't know the full term of it. It's like SATA something something. It's like 1,000 letters. And then
Yeah.
A is stands for androgen receptor. So
Okay.
it's like a basically like like androgen is like a protein that's like
Testosterone,
associated
right?
with like, yeah, testosterone, stress, etc. Basically
Yeah.
like emotion in a way. Basically like androgen receptors, or inhibitors in some case, are basically um, like you said, testosterone, as well as other like hormonal things. Like I
Mm-hmm.
think estrogen is also carried by androgen receptors too. Um,
Mm.
so that's like the nomenclature to an extent. But basically, those proteins are hallmarks of these cancers. And our his goal, which is now I guess our goal in a sense, because we're working on creating basically a model for this, is that if you can detect these early enough with a biosensor,
Mm.
you can basically detect them to the point where it's so much earlier than any other cancer detection that it's almost like
Mm.
you don't actually have the cancer yet, right? Because now we detect cancer through like mostly through tumor genesis, right? And once you have
Right.
developed
Once you
tumor
have a
genesis,
tumor.
yeah, once you have tumor genesis, the cancer is basically moving in a way that you can really only try to slow the train down rather than stop it completely.
Mm-hmm.
So that was his research prior to coming to St. John's as a new faculty member.
Mm-hmm.
And so I'm in his lab now, and I am we're working on this project together to get it off the ground floor. And then from that point, I think that my individual contributions, or what we get from that, will kind of lead to my dissertation as a whole. I would
Right.
love to personally start to get into the RNA world of that, as
Mm-hmm.
my master's degree, et cetera, got me into RNA. That was what my master's thesis was on. It was like cavefish RNA. So
Cavefish RNA.
yeah, I'll get into that in a second, but just so I don't lose my train of thought. But I would love to get into the RNA development of these proteins, as
Mm-hmm.
well as hopefully doing some sort of reverse engineering to create DNA back out of these RNAs, and then kind of like see um, what part in the body they come from, where in the genome they're localized, et cetera, what
Mm.
other proteins are synthesized from that locale in the gene, and perhaps what we can learn about transcription factors or anything like that that latch onto that area of the genome, and any deficiencies you may find common throughout disease and stuff like that. So we're currently still in ground stages. We've been, a couple months. These things take time. Progress is a uphill battle a lot of times, a
Sure.
lot of repetition in science, as I'm sure you know. So we've been able to find full sequences actually for all of these proteins, these 4 proteins we're looking at. We've cut it down to
So
two
how did you
proteins
find
right now.
sequences for them?
So basically we just my first task was basically to like pore over literature about them, find anything you can. Um,
Mm-hmm.
and this was actually one of my first, I would say probably my first experience in poring over scientific literature in a way where I wasn't finding 1,000 results that were cited by 1,000 different people, right?
Mm-hmm.
Because a lot of times when you're in an academic situation, or even when you're doing a master's thesis, which is a more guided program, right?
Mm-hmm.
Any proteins or genes that you're looking at or have been told to look at are oftentimes very popular, and oftentimes
Mm.
ones that have been studied very many times. And if you type their name into Google Scholar, you'll get 10 great results from very great accredited journals that have
Right.
all been cited upwards of 1,000 times. Um, Whereas, right, so if you did type GFP, as we mentioned before, into Google Scholar, you are going to get, your first 20 pages are probably going to be all publications in great journals like Science or Nature or the
Mm-hmm.
New England Health Journal, and they're all going to be cited 2,000 times, and they're going to be very well established. Whereas these 4 proteins that I was looking for, very little literature on them, very like obscure, and that's because they were like, they had to be obscure in a way, right? Because they were originally picked by my PI because of the fact that they are both hallmarks, and they're also important, also just as importantly, not particularly present
Mm-hmm.
in other parts of the human genome, right?
Mm-hmm.
Because if this pip high protein, for example, gets produced every time you like ate a slice of pizza, it wouldn't be a
Right.
particularly great marker for
No, it'd be
something as
useless. Yeah.
serious as cancer, and probably
Right.
as expensive and time consuming as getting a mouthpiece with that
Imagine
biotechnology
the
in it.
doctor's like,
Yeah.
"Oh, you have cancer," but it's actually you just ate pizza.
Yeah, yeah. Oh well, look, it says here you're producing this protein, and it could either mean 2 things: you're dying, or you had a large breakfast. And
Ah,
it's like,
damn, I can
right?
never
Yeah,
remember what I ate for
yeah.
breakfast.
Exactly, right. So those are the type, that's hyperbole, but those are the things we want to avoid. So
Right. So
as
those
I
are
was,
usually only produced if you have oral cancer.
yes, right. So they're kind of obscure proteins, and so I spent a while looking through research so I could find some valid stuff. And what we ended up coming down to was we ditched one of the proteins because it was something that had only been talked about in a single paper published in a journal like 10 years ago, some like ad hoc journal about like alligators, something
Mm-hmm.
like that. It was like found in alligators and named that way. Um,
Okay.
and actually, that's how it got its name, because it's MRA, and it's from the American alligator, and it was also an androgen receptor.
Mm.
Um, so uh,
Not a particularly close relative of ours in terms of
no, pretty
evolution.
pretty far as most of the extant.
Yeah.
Yeah, that's probably about as far as you get actually with all the extant organisms today. Um, but that said, so we cut it down to 3 proteins now we're looking for. And then once you get the protein, so once you have literature on the protein, basically what you can do is find the sequence of it, right? So my first goal was like find sequences of amino acid sequences of these proteins. And then once you have the amino acid sequence, it's easy. You can use you can use a database like NCBI or
Mm.
XPACY, which are 2 databases. Funded like national databases, international databases too as well, NCBI at least, where there's tools where you can input, reverse engineer or engineer a sequence based on having the RNA, DNA, or protein sequence, right? So if you have the amino acid sequence of something, you can put it into one of these databases and it will spit back out the DNA sequence that came from it, because all you have to do is reverse 2 steps, right? Because
Yeah.
RNA
So
is the transcript for DNA, and then the protein is synthesized from the RNA transcript.
I think if you could kind of describe that, in the hope that one day we'll have lots of listeners,
Oh,
that
sure,
I'm sure some of them won't really know the relationship between RNA, DNA, and protein. And since you've spent so
sure.
much time teaching Bio 1, could you
Yeah,
briefly give us the outline?
I would love to, Dan. So what I actually just described is called the central dogma of molecular biology, and what that means is you start with DNA, you move to RNA, and you move to protein, right? So that's simple enough. These 3 steps. What that means is, right. So all of these organisms on Earth, yourself included, you have these strands of DNA. And all the proteins that create all the cells and every function and every motion and thought and movement and signal and hormone you
Right.
create,
Proteins do like everything, right?
yes, are either developed by proteins or are a protein themselves, or they're a
Mm-hmm.
mass of proteins together. So the DNA is kind of like, think of the DNA as a blueprint, right? You're
Mm-hmm.
building a house, you got a blueprint. Um, and then the RNA, right, is kind of like the transcript of that DNA. So your RNA is like your materials for your house, right? You've got a blueprint, and then based on the blueprint, you spit out some materials for that house. And then the protein that builds from that RNA, a protein is a simple word for a building block of several amino acids. There are 20 amino acids, and RNA codes for some or many of those amino acids, and then those amino acids as building blocks create your protein, or your house in this case, right? So what I'm saying, to relate that back to what I was saying before, if I find the amino acid sequence of this protein, right, I can then use a database, which has just the general information that I was just talking about, where if I have a sequence of amino acids, I can plug that into this database and it shows me the sequence of RNA that would spit out that sequence of amino acids, right,
So
as a
I never knew
transcript.
that you could do that reverse process,
Yeah,
because
you
I kind
can.
of figured that like more than one RNA sequence could code for a particular amino acid or something, but that's where I get a little bit
Yeah.
foggy.
Well, so that's interesting. It's so there's things called tRNAs, transfer RNAs, which are
Yeah,
basically what puts the specific amino acid codes
right,
together. And every tRNA, right? So every RNA piece specifically corresponds to one amino acid, but
right.
an
And they're
amino
what,
acid,
3 codons long or so, three
yeah, they're
amino acids?
three amino acids long for each tRNA. That they synthesize an amino acid, but the codons of the mRNA transcript, basically, you have these transfer carriers RNA, and they create those building blocks of amino acids by each single tRNA is specific for an amino acid,
Hmm.
but each each amino acid is not specific for an RNA.
Right.
So you can basically say that, like, if I have an alanine, right? Let's say
Hmm.
part of my protein subject is an alanine or an A, and that's in my sequence. I know which codon of RNA brought me that alanine,
Hmm.
right? Um, my dog keeps jumping up here. So I know
Yeah.
which codon of the RNA brought that alanine to my building block. And
Hmm.
so what I can say, okay, I have an alanine, which means that I have an a G, or a U, right?
Hmm.
That's just made up, but that's what that's saying, right? So like my carrier for that was an A, G, and a U.
Hmm.
And then from there, you can the mRNA, RNA is just DNA, but where uracil is replaced for thymine.
Right.
So
So
you
if
can
you
do
know
the
the
same thing.
RNA, you can figure out the DNA. It's very one to one.
Exactly,
Yeah.
it's 1 to 1. Um, so so that's basically what I did once I found the amino
Yeah.
acid sequences. You can put it into these databases and quickly find the RNA sequence, and then from there it just brings you straight to the DNA sequence because it's a like 1 to 1 aspect.
So you did that for these sweet proteins?
Yes.
Okay.
So I did that for these proteins, got the sequences, and then from there we have moved on to the sequences where we're trying to create primers for them now, because
Mm.
we basically want to not expand. What's the word I'm looking for? Uh, Amplify
Mm.
these regions that we have, right? Um, So once we amplify those regions, we'll have a lot of them to work with. And then we're going to our end goal here with this specific part of this project is create a bunch of the DNA that we need that corresponds to those specific proteins. And what we're going to
Mm.
try to do is link them through a linker gene to an operator in a plasmid, which is basically, think of it as like your little, like, it's a little cloning device
Yeah.
that's isolated from a bacteria most of the time,
Yeah.
a bacterial DNA. And because of that, you can basically um, you can insert whatever you want into that little circular piece of
Hmm.
DNA and clone it.
It'll
And
replicate,
so
right?
that's our first step here, is right. We want to clone a bunch of those pieces so that
So that you want to
eventually,
clone the DNA that creates these three proteins, right?
yes. So
Okay.
we want to
So
clone. Thank you for synthesizing that. We want to clone the DNA that creates those proteins multiple times. We want to amplify it so that we can eventually, so that what we can end up doing is get to the point where we have the ability to have them linked to our linker gene, which is going to be something called our reporter gene, right?
okay.
So that eventually in this engineered biosensor, when this protein is linked to is basically synthesized, our
Hmm.
linker gene will basically create a synthesis of a reporter, which will say, ding, 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 protein synthesized problem.
Okay. So the
Yeah.
biosensor's job is to detect one of these three proteins, right?
Yes. If or
Um,
all, ideally.
do you know that these protein, like, do you already know that these proteins are only made when the person has oral cancer?
So we know that they're hallmarks, and we know that they're very rare. We don't know 100%. We can't say 100% that they're only synthesized by humans with oral cancer.
Right.
But that is also something we want to look into. I'm not sure how we're going to do that yet, or how my PI really wants to proceed with
You'd
that.
have to do
But part of
human subjects work, right?
probably mice first and then human
Yeah,
subjects
right.
eventually.
Right.
But the goal for also amplifying a bunch of these genes is not to just link them to a potential biosensor, but to basically like explore if we can have them get produced in any other way, right? Like we want to
Okay.
integrate them into a DNA. You want to integrate them through a plasmid into a specific organism. And then once it's
Mhm.
integrated into a specific model organism's DNA and gets synthesized, you can see if it's naturally synthesized in another way. And I'm not very when it comes that's where my expertise is not
Mhm.
particularly heavy, so I'm not going to keep talking about it like that. But I know that that's one of the things that is crucial when you're doing anything as far as like some sort of public health
Right.
um, technology is concerned.
Yeah. All the stages of
Hmm.
trials to make sure that it's effective. And I think the times we're living in now make biology feel so much more relevant than it ever has, right?
Hmm.
Not that it wasn't relevant before, but like my current work is mostly working in schools and tutoring. That I'm currently I just tutor. Everything's
Hmm.
online. And it's like people who used to be like, well, it's like one of those sound bites, right? Of like what school teaches you as an example of something useless. They would always have
Yeah.
like the mitochondria as the powerhouse of the cell, right?
Yeah. What does
Um,
that mean though? Yeah.
yeah, and it's like when I remember when I first heard somebody make that point, it kind of hit me hard because I was like wanted to be a biology teacher, right? And it's like
Hmm.
I don't want to do a job that's useless and like teach kids things that is not useful. But
Sure.
this pandemic has made me realize how useful that knowledge can be. Um, I mean,
Hmm.
especially for somebody who wants to go into research or medicine, right? But even for the average person to try to understand what is an mRNA vaccine, for instance.
Hmm.
Um, so
Yeah,
that's
it's
what I tell
not.
my students now. Like, if you think knowing about the cell is not important, just think about what we're living through now and how it has to do with the medicines that we use. And
Hmm.
if you really want to know what's in that vaccine, you have to know what mRNA is. You have to understand that it's how proteins are made and all that stuff. So
Hmm.
it is
Absolutely.
interesting.
And if you don't, if you just know it at a baseline level, it's not really going to give you information you want. It also leads to things where people say here mRNA vaccine, and they get to
Oh yeah,
read a conspiracy theory and immediately believe it because it's easier to understand than something else.
that's true. Sometimes knowing a little bit about something is worse than knowing nothing because you just
Yes,
misunderstand it.
yes, absolutely, absolutely, dude.
But
um
do you know much about how mRNA vaccines work? I think
I
our friend
do, yeah.
Adam Quick was telling me. But so mRNA, right, is like the DNA
Yeah, let me
is
put this
essentially
charger in.
translated, and the process is called transcription, right? Which is
Yes.
interesting. But it's like the DNA code is translated into an RNA strand, right?
Hmm.
The RNA strand then floats out of the nucleus over to the
Hmm.
ribosome, and that's the
Yes.
mRNA, right? The messenger RNA. And so these mRNA vaccines are actually strands of messenger RNA, right? That
Yes,
they
they are.
inject into you. So it goes into your cells, right?
Hmm.
It goes to the ribosome and it tells the ribosome to make certain protein.
Yeah.
Is that right? So
Yeah,
it's
I
actually
mean,
gets your
that's
cell to
basically
make certain proteins.
it. That's you said it pretty well in plain language. The only thing I will correct is it doesn't tell the ribosome is not told anything. A ribosome is like a structure of
Mm.
RNAs that just kind of holds your transcript.
Yeah, that's right.
It's
The
like
ribosome is made of RNA itself, right?
the ribosome itself is a big pillow of RNA that kind of is just like
Oh.
a little table for all the like decoding to be done.
Mm.
It's like it holds things in place and transports. It's your basically it's your vehicle for translation.
Mhm.
is what I like to say about it. So yeah, I mean, Dan, you nailed it pretty well. The thing about an mRNA vaccine and why they're pretty novel, but they're great because in a case like this, we can use basically a single spike protein was isolated from the SARS-CoV-2 viral
Mhm.
particle, and
That's spikes on the outside of it, right?
spikes on the outside, right? So it's from our not from multiple months of amazing research by some amazing scientists, as well as previous knowledge we have of SARS coronaviruses, um, previous to COVID-19, scientists were able to um, identify this spike protein from which an mRNA was reverse engineered.
Mm.
And
Just
that,
like kind
remember,
of you did with your
ex
proteins.
exactly, right? It's the same basic concept, right? We have this protein, you break it down to its essentials. Once you've broken it down to its essentials, you can basically say, okay, this amino acid here means that I have, let's say I have a serine, right? So
Mhm.
UCA here, then I have a leucine, CUA, right? So you say those things, then what you do is you basically have an mRNA transcript, that you can, you have a blueprint for it, that where you can develop artificial mRNA, um, to inject. And the reason why it's safe and not why it's much different than a traditional, like the first vaccines for like a polio, for example, right? That was
Yeah.
injecting a little bit of the piece of the dead virus, right?
Yeah.
This is completely different. You're not injecting any of the virus. In fact, everything you're injecting is synthesized. You're not injecting mRNA that came from people with COVID. You're not injecting mRNA that came from an actual COVID particle. It is
Mhm.
simply a engineered strand of RNA that is put into a vaccine. And once it gets injected into your body, what happens is it gets integrated into your genome. So what happens is that mRNA is now in your body. So that mRNA is going to dictate your body to naturally produce a protein that is not a SARS-CoV-2 protein in general. But it's a protein that is very similar to it, right? Because it's a piece of that spike protein. And once
Okay.
your
So
body
you're not
naturally,
creating the spike protein actually, you're creating something like part of the
you,
spike protein.
I mean, it's kind of like it's like if the so if COVID itself is mu, right? The protein you're creating is mu2, right? It's
Thank
not
you
as
for that
the
explanation.
powerful, the power part aside, right? It's kind of like a synthesis. It's a lab-based synthesis based on
Yeah.
the original, right?
Yeah.
So your body now with this mRNA inside of it is going to naturally produce a protein similar to a COVID
Right.
protein. And what that does is stimulates the immune system to create antibodies based on
Right.
that antigen receptor, so that if and when, God forbid, you are exposed to
Right.
a sickness dose of COVID particles, your
Mhm.
body is already familiar enough to the point where your immune system does not integrate it, because it
Mhm.
already has the ability to cut it out of your genome before
Mhm.
the viral particles take over uh, production.
Yeah, so I've been doing a little reading up on the immune system lately, and if you really get into the detail, it's like mind-boggling about antigens and antibodies, and like there's so many like the parts of the white blood cell that actually attach to the foreign body, and it's like they recognize part of the foreign body and stuff like that. So usually,
Mm-hmm.
right, the general explanation is just that the foreign body is recognized by white blood cells
Yes.
that will then attack anything with that foreign body on it. So in this case, the piece of the spike protein, right, which
Mm-hmm.
is on coronavirus cells, that the white blood cells will know to attack those because they've seen it before, right? And so you have
Yeah.
basically,
Yeah. It will basically, it's not, it's T cells like white blood cells too, but I think it's like it's other like endonuclease activity, which endonuclease activity is
mm-hmm,
a fancy word for basically your body's proofreading system, right? If it sees something in it, like a foreign piece of DNA,
mm-hmm,
your endonucleases should cut it out. And that's
mm-hmm.
what this is priming the immune system to do, right? Like T cells
Oh,
may like digest it, cut it, whatever it is that I don't know specifically what it does, but whatever it
yeah,
does, it basically says, hey, we have recognized a protein with this exact sequence before, right? So when this viral particle is now trying to hijack your own machinery, endonuclease activity will basically bonk it and say, no, you go no further here.
Yeah.
And then mRNA in general, again, is degraded, right? So once an mRNA transcript has done its job, it doesn't hang around, it gets degraded.
Right. So it's not, I've
And
heard the conspiracy that this was like changing people's DNA, but it's
no,
not. It's not even DNA, it's
has nothing to do
mRNA.
with DNA. There's
Right.
there's
Well, it has
no,
something to do with DNA, but it's not
yeah, it's
DNA.
not DNA. You can inject any mRNA of anything. It will never, it might hurt you in some way because of whatever you're putting in, but it will never, your body does not reverse engineer DNA from RNA. That is strictly a
Right.
laboratory technique. The body does not do it.
Well, Sam, I need to
Yeah,
cut out of this conversation now, but I think we touched on some good stuff. So,
I think we did. I
as
think
our
this was
final
great.
word from an expert, should people be afraid of the vaccines?
I'm glad you asked me that. It's a great final word. So, my answer to this, and my simple answer is no. You shouldn't be afraid. My longer, more advocacy and public health and bioethics answer is I understand if you are. Um, our country does not have a good track record of public health, healthcare in general, let alone advocacy when it comes to vaccines, especially for Black and Brown people. Um, we are less than 100 years out from something called the Tuskegee experiment, which was a very high profile CIA experiment to basically give Black men syphilis to try to study it. It was completely un
Unethical.
it was unethical in every sense of the word, and not it was classified information until probably like 10-20 years ago now. And to that, I understand any type of skepticism, especially if you don't have the knowledge base that, or a college education or even high school education. Sometimes I get it. And I don't think that shaming these people or making fun of these people who might be afraid or whatever is a way you should go at all. I think we need strong, educated, meaningful, non-pandering advocacy to get us out of this, to convince people that this is something that they should feel pride in getting and that they should feel safe in getting. And I think that we are butchering that as usual. And I think that the general way that this vaccine has been administered with no seemingly rhyme or reason across state lines is only hurting the case. And I think that now that even more so, we're going on almost 2 months of this vaccine, the first person in America getting this vaccine and public being able to get it. And, with the virus still raging, I think you leave the door wide open for people to say, I told you so, it's not real, this is a hoax, etc. And so, I won't keep going about it. But again, my short answer, no, you should not be afraid of this. My long answer, I understand if you are. And I apologize on behalf of the scientific community for the aloofness that many times many of us are involved in, that does not, it in no way opens the door for understanding from anybody outside of the club. And
so a call for more
a call for advocacy. My answer to you
advocacy
is
and
a very strong,
education,
yes, a very
communication.
strong call for advocacy, a very strong call for public health integration, a very strong call for understanding in bioethics.
Cool.
Yeah.
Well, we're spreading knowledge with the
Yes.
podcast.
Spreading
So
love and spreading fucking knowledge, dude.
spreading everything except
Spreading those
COVID.
cheeks for me.
We spread everything but COVID here.
Facts.
But yeah, so that's going to be the end of our convo today. Sam, thank
Yes.
you very
Thank you.
much.
I love you,
Talk
Dan.
to you
Thank you
in
for
about a
having me
minute.
on. Yep. Bye, dude.
Bye.
Have fun with your bro.
We'll do our best.